Welcome to this lecture on local and global alignment in computational biology. Imagine you have two slightly different versions of a recipe for baking a cake. If you want to see exactly what changed, you would place them side by side and match up the identical steps. In computational biology, we do exactly this. But instead of recipes, we are comparing long strings of biological letters, like DNA or proteins. This process is the absolute foundation of bioinformatics, and today we are going to master how it works. Now that we understand the basic idea of comparing recipes, let us look at how it applies to biology. Formally, sequence alignment is the process of arranging two or more biological sequences, such as DNA, RNA, or proteins, to identify regions of similarity. Why do we care about these similarities? Because they often indicate crucial functional, structural, or evolutionary relationships between the sequences. To make the sequences line up properly, we are allowed to insert gaps. These gaps are not just mathematical tricks, they represent actual insertions or deletions of genetic material that have occurred over evolutionary time. Our ultimate goal in any alignment is to maximize the number of matching characters while minimizing the number of gaps and mismatches. This goal of maximizing matches leads us to an important question. Why does this matter in the real world? Imagine a scientist discovers a brand new gene in a fruit fly. Initially, they have absolutely no idea what it does. However, if they align this new sequence against a massive database of known human genes and find a perfect match, they can instantly predict that the fly gene has the same function as the human gene. Without sequence alignment, every new genetic discovery would remain a complete mystery. We would have no way to compare the genomes of different species. Today, it is the primary tool used in drug discovery, evolutionary biology, and understanding genetic diseases. Having seen the real-world impact, let us dive into the first major type of alignment, known as global alignment. Imagine you are comparing two different editions of the exact same book. You want to stretch them out and compare them entirely, forcing every single word to align from beginning to end. Global alignment does exactly this with biological sequences. It attempts to align every single letter of the first sequence with every single letter of the second sequence. The most famous method for this is the Needleman-Wunsch algorithm, which uses dynamic programming to find the optimal alignment. Because it forces an end-to-end match, it is most effective when the two sequences being compared are of roughly the same length and are highly similar overall. Now that we understand what global alignment is, let us look at how we mathematically score it. The formula you see calculates the total final score of the global alignment, represented by S global. This score comes from adding up three components. First, we have N match, which is the total number of matching characters, multiplied by P match, the positive points awarded for each match. Next, we add the mismatches, multiplying the number of mismatched characters by their specific penalty score. Finally, we account for the gaps. N gap is the number of gaps introduced, and P gap is the penalty for each gap. If you increase the gap penalty, the algorithm will try harder to avoid inserting gaps, which might force more mismatches. The goal is to find the arrangement that makes S global as large as possible. Before we move on, let us see this scoring formula in action with a concrete example. We are given two short DNA sequences. Sequence A is GATTACA, and sequence B is GATCA. We need to calculate the global alignment score. Our rules are +2 for a match, -1 for a mismatch, and -2 for a gap. First, we line them up end to end. Since sequence A has 7 letters and sequence B has 5, we must introduce exactly 2 gaps into sequence B. We align them so the first 3 letters, GAT, match perfectly. Then we insert 2 gaps to align with TA, and the final CA matches perfectly. Counting this up, we have 5 matches, 0 mismatches, and 2 gaps. Plugging these into our formula, 5 matches give 10 points, and 2 gaps give a -4 penalty. 10 minus 4 gives us a final score of 6. This positive score indicates the sequences are quite similar. Having successfully calculated a global score, let us imagine a completely different scenario. Suppose you are looking for a famous quote hidden somewhere inside a massive encyclopedia. You do not care about aligning the entire encyclopedia to your quote. You only care about finding that one specific matching paragraph. This is the exact intuition behind local alignment. Instead of forcing the whole sequence to match from beginning to end, local alignment searches for small, highly similar regions hidden within larger, potentially unrelated sequences. It is like finding a needle of similarity in a haystack of genetic code. This approach is incredibly powerful when you are comparing sequences of vastly different lengths, where global alignment would fail by introducing too many gap penalties. To achieve this targeted search, we use a different mathematical approach. The standard method for local alignment is the Smith-Waterman algorithm. Unlike global alignment, which is rigid, this algorithm allows the alignment to start and stop absolutely anywhere within the sequences. The mathematical trick that makes this possible is brilliantly simple. The running score is never allowed to drop below 0. If a region of the sequence is completely unrelated and the score becomes negative, the algorithm simply resets the score to 0 and starts looking for a new matching region. By cutting off bad alignments early, it isolates only the highly similar local regions, allowing us to find shared functional domains in otherwise unrelated proteins. Now that we understand the Smith-Waterman trick, let us look at its mathematical engine. This formula represents the core decision made at every single step of the local alignment algorithm. The variable Hij is the alignment score at the current position in the 2 sequences. The function max means the algorithm will evaluate 4 different options and choose the highest value. It checks the score for a match or mismatch, and the scores for opening or extending a gap in either sequence. But the most critical option is the 0 at the very beginning. This 0 means that if all other alignment options result in a negative score, the algorithm will just pick 0. It effectively cuts off the bad alignment and starts fresh, preventing negative scores from dragging down a good local match. Let us apply this local scoring rule to a scenario where global alignment would fail. Suppose sequence A is a long gene, ATGCGTACGTTAG, and sequence B is a short fragment, TACG. We want the best local alignment score, using +2 for a match, -1 for a mismatch, and -2 for a gap. If we used global alignment, we would have to add 9 gaps, resulting in a terrible negative score. Instead, we slide sequence B along sequence A until we find the best matching subregion. We find a perfect match hidden in the middle. Because this is a local alignment, we simply drop all the unaligned letters on the left and right. We do not penalize them as gaps, we just ignore them. We have exactly 4 matches, which gives us a final score of 8. This perfect local match we just calculated brings us to a visual summary of the 2 techniques. Look at the diagram on your screen. In the top section, representing global alignment, you see 2 sequences of similar length being compared from the very beginning to the very end. The dashed lines span the entire width, showing that every single character must be accounted for, even if it means forcing gaps. In the bottom section, representing local alignment, you see a much shorter sequence finding its perfect home in the middle of a longer sequence. The dashed lines only exist in that specific subregion. The rest of the longer sequence is completely ignored. This visual perfectly captures why we choose global for overall similarity and local for finding hidden domains. We have covered a lot of ground today, so let us summarize the most important ideas. First, sequence alignment is the mathematical process of comparing biological sequences to uncover hidden evolutionary and functional relationships. Second, when sequences are similar in length, we use global alignment, powered by the Needleman-Wunsch algorithm, to force an end-to-end comparison. Third, when we are looking for small matching domains within sequences of vastly different lengths, we rely on local alignment and the Smith-Waterman algorithm. By understanding how to manipulate match rewards and gap penalties, you now have the fundamental tools needed to analyze evolutionary relationships and discover the functions of unknown genes. Great job following along with these core bioinformatics concepts.